Hi, my name is Lisa.
My name is Sahra.
We're from Ishik Nilufer school. So our project is about antibacterial effects of green synthesized nanoparticles using Plavra against the Staphylococcus aureus. So we are going to have a plant, so we can consume it as food. This malva plant, we can consume it as food. It's been very in our culture, and we progress some tests on it. We progress some tests on it and make a good antibiotic. And the results showed many advantages. So we took the malva plant as a fresh form and boiled it in the lab and made the accuse from it. We biosynthesized the silver nanoparticles with the malva and evaluated the antibacterial efficiency against the Staphylococcus aureus. The Staphylococcus aureus is a type of bacteria that is found in the human nose, about 30%. It is very effective and it's grown positive. So here we have a petri dish. A petri dish, so here the first one we have is a simple petri dish called the Mitz agar. And the second petri dish is called the Muller agar. We put the malva, accuse, and bacteria inside it. As you can see, there has been no bacterial growth. But here we put other antibiotic inside, not malva, with bacteria, and there has been a lot of bacterial growth. And now my cousin can continue.
The final result of our test on malva medicated with silver nanoparticles showed an effective antibacterial agent which stopped the resistance growing against the antibiotic for the bacteria. Unlike other antibiotics, it's a good alternative for bacterial control. As a result, it works better on urinary tract infection, skin infection, strep throat, and it works better than vancomycin for colitis.